Hi everyone. Welcome to this seminar series of the Center of Excellence of Youth Mental Health. So it's a great pleasure to introduce the speaker of this month, Dr. Paul Arnold. He's the director of the Mathison Center for Mental Health and Education and the Alberta Innovates Translational Health Chair in Child and Youth Mental Health at the University of Calgary. He's also a professor at the Departments of Psychiatry, Cumming School of Medicine at the University of Calgary, and a child and adolescent psychiatrist practicing at the Alberta's Children's Hospital. And his research focuses on how genetic and environmental risk factors interact to develop the development of child onset neuropsychiatric disorders, early interventions in child and adolescent youth mental health, and he has extensive experience leading large programs integrating research and innovation with clinical care. Two key examples of leadership include working with Alberta Health Services to develop new research-intensive community-based child and adolescent mental health center in Calgary and transform mental health services for emerging adults in the Calgary zone. So today he's going to present some of this important work he has done on OCD, genomics of obsessive compulsive and related disorders, the path to personalized medicine.
Great. Yeah, thanks very much, and thanks very much for coming. So nice to see all of you here, and also just shout out to those of you who are online as well. As Linda mentioned, I'm going to be talking about genomics of OCD, let's make sure I can advance my slides. Okay, that's why. Got it. Okay, thanks very much. So yeah, here are my disclosures. Not really directly related to this presentation. So the only commercial disclosure we have is we do have a pharma sponsored trial, but that's won't be talked about today. So I'd also like to begin by acknowledging that this talk is being given from the traditional territories, and again, apologies in advance for any pronunciation errors. But the Ganekiaka, and it's also the traditional site of meeting and exchange among the Haudenosaunee and Anishinaabe nations. And I'd like to give a sort of an overview of what I'm going to talk about today. So I'm going to sort of start by orienting you a little bit to both psychiatric genetics, a little bit to OCD as well, just in terms of just basics of OCD, diagnostic criteria, very, very quick, just to ground us. Then going to go, probably the bulk of my talk is going to be focusing on the genomics of OCD, some of the research we've done using kind of a variety of different approaches on the genetics of OCD, but also starting to think about how we might link it to other kinds of risk factors, particularly environmental risk factors. And then I'm going to talk about it, and this is very much aspirational around, so I sort of entitled the talk maybe somewhat ambitiously is about kind of the path to personalized medicine. We're not there yet. We're very much on the beginning of that path, but want to just talk about some directions we could go in, give some concrete examples that are sort of adjacent to the work we're doing in OCD, where we're working in pharmacogenetics, and maybe how we can build on that to to really achieve the goal of a lot of this work, which is to develop more precision medicine approaches. So again, this is just very high level. And I'm, kind of when we think about the etiology of mental illness, sometimes we like to think about it from the genetic code to the postal code. So it's, it's not just genomics is where I'm going to spend a lot of this talk. So genetic risk factors are really important. So for many of there's variability, as you'll see, in terms of the heritability of psychiatric disorders. But certainly, as in the case of OCD, for example, there's approximately 50% heritability. So it means there's a strong genetic component, but of course that's influenced by the environment, including, for example, social determinants of health. It's things you can determine not only from factors that happen in your family, like experience of early trauma, for example, but also things that may happen. And one of the things we're looking at some of our data, and I'll just touch on it very briefly, is just sort of neighborhood level factors, like what kind of environmental context did you grow up in particular, can really have an influence on the later development of mental illness and physical illness as well, but certainly mental illness. So kind of coming back to genetics, so in terms of heritability, again, we know that most of the disorders that we're dealing with, and I put up here for illustration some of the disorders I've most of my work has been in OCD, but also more sort of being involved in kind of collaborative studies looking at the genetics of ADHD and autism. You can see some variability. So some of our neurodevelopmental disorders have a higher heritability. ASD, autism spectrum disorder, very high. OCD kind of falls a little bit in between, and because and not on this slide. Major depression, for example, has a somewhat lower heritability, but certainly there's a substantial genetic contribution, and that's kind of the starting point why we're doing genetics in the first place. There's a long history, very much predates some of the more modern genetic approaches I'm going to talk about, of doing things like family and twin studies. We have lots of evidence that these disorders run in families and that there's greater shared risk amongst identical twins versus fraternal twins, that sort of thing. I'm not going to talk a lot about those studies, but that's sort of in the background of this. We know these are strongly heritable. And another sort of point of sort of orientation is like, so now that we're shifting from understanding heritability to looking at the actual genetics of these disorders, what approach do we use? And there's this kind of within the field of psychiatric genetics, there's these two different approaches of either focusing on rare and so I'm actually going to use my pointer because then folks online can see this, my pointer on the computer, so folks online can see this as well. There's rare variants that are considered what we call highly penetrant. Penetrance means just if you have this variant, how likely it is you're going to have the trait or phenotype of interest. So a highly penetrant variant means that, an extreme end would be something like if you have the genetic predisposition to Huntington's disease, you're going to get it basically, but it may depend on when you get it and so forth may vary, but you're actually going to get the disorder if you have that risk gene or that risk variation. If you're however, if you're more, what a lot of the work that we do in psychiatric genetics, and there is work in the rare genetic, and I'll touch on it very briefly. A lot of the what I'm going to focus on, though, is more on the common variation. So if we did a survey of this room for any single one of those variations, you'd find like a very high prevalence. There'd be somebody in this room who has this genetic variation. And so alone, it's a low penetrance. So it means that, but it doesn't necessarily mean that person has, let's say, OCD kind of thing, if it's the variant, the risk variant for that disorder. But if you have we think that some kind of complex polygenic combination of many of those variants may then predispose you, increase your risk or probability of having this disorder or trait of interest. And the sort of the genome-wide association studies, for example, are very much premised on this common disease, common variant hypothesis, that the common diseases are the result of multiple common variants of small effect, and that tends to lead itself to that genome-wide association approach. And then there's also this concept of like, well, there could be multiple rare variants that lead to that. Individually they have large phenotypic effects. Individually they're rare, but if you pool them together, they count for a burden of disease in a population. So that lends itself more to copy number variant or next generation sequencing studies, which are the types of techniques that are used for that. And I'll touch just a little bit on that, but focus a bit more on the GWAS approach. And it's also important. Another key principle I wanted to point out is that your DNA doesn't read the DSM-5. So it doesn't, doesn't really respect diagnostic boundaries. And again, this is consistent with the trend in our field, like with the RDoC approach that is promulgated by the NIMH, for example, that we think that that really there's a lot of transdiagnostic risk, and that's very, very much true in genetics. So, and there's been some kind of work pooling together some of these genome-wide association studies. And I'll touch on that as it applies to OCD, suggesting that there's common risk across multiple disorders. And another principle is that, size matters in genome-wide association studies. And we'll talk about that, how OCD is starting to just hit the critical threshold where we can actually find things. And this is basically a slide from Patrick Sullivan, who's one of the leaders in the Psychiatric Genomics Consortium, published a few years ago, but it's very much true. The actual exact figures have probably changed a little bit, but the principle is there. So on the x-axis, you have the number of, so if you do a genome-wide association study, what you're doing is you're looking at multiple variants across the entire genome. And your hope is that you're going to find not one, because maybe that one may not replicate, but multiple significant loci that you can then follow up with other approaches. And on the x-axis, we have the sample size of the studies that have been done. And you can see here that, and another piece that's important is the heritability, which can be measured on twin studies. There is also a way of measuring what we call SNP heritability, but which is actually looking at the genetic variations themselves and how they contribute to heritability. But the more traditional metric of heritability is twin studies. So You can see here for the disorders that where genome-wide associations have kind of broken through in, let's say, the last 5 years, really started to find things are like bipolar disorder and particularly schizophrenia. But that's that kind of combination of schizophrenia is thought to be highly heritable, and not thought to be, but lots of evidence is highly heritable, and also they have huge sample sizes because of combined efforts, well-resourced across the world, and so they're crossing that threshold where they're finding lots and lots of things, and they're now trying to figure out what it all means and trying to follow it up. But whereas for some of our other disorders, OCD, it's shifted, by the way, as you'll hear. But at the time that this slide was, but OCD was kind of in the mid-range in terms of heritability, but sample size is way too small. So we weren't finding things. We'd only found at that time, we'd only found one locus kind of thing. So really kind of interestingly, we're really in the far left of this. So as you're here, we're starting to break through that. Tourette syndrome, similar. And actually, there's been a lot of common pathways to find the genetics for those too, but a bit more heritable. And then we've got major depressive disorder where sample sizes have actually been large, but it's been harder to find things because there's thought to be a stronger environmental contribution. The heritability is actually lower for major depressive disorder. So it's just more challenging in that regard. So just wanted to ground you but I also in terms of foundational ideas for this talk, I also just want to talk just a little bit. So I'm going to focus, I've talked in general about psychiatric genetics. Much of the talk is going to focus on OCD. So I thought I should just say, what is OCD for, kind of thing. And many of you will be familiar with it and the diagnostic criteria, but just to kind of, set the stage for that. So obsessive compulsive disorder is a disorder of that comprises either obsessions, which are intrusive, repetitive thoughts that typically the person recognizes unreasonable or senseless, and that recur over and over again. So an example, if I was really concerned about contamination, that sort of thing, germs, I might be having a hard time focusing on this talk because again, it's intrusive. It's getting in the way for me. Compulsions are repetitive behaviors that one has to do over and over again, either in response to an obsession or sometimes it's just a feeling of anxiety, just a more general sense of unease if you don't do these acts. So you don't actually have to have both obsessions and compulsions, although majority of individuals do. And again, the correlate to that contamination was, the one you'll hear about a lot with OCD is I had to wash my hands excessively or not touch things, for example. That's sort of an avoidance behavior that's like a compulsion. These are time-consuming or cause clinically significant distress or cause impairment in social, occupational, or other important areas of functioning. So that's pretty similar to other criteria. Interestingly, there's no duration criteria for OCD. So, there's not actually in the DSM a sort of length of time that it has to occur. And then the other kind of rule outs are very similar to other DSM disorders. So not due to direct physiological effects of a substance or another medical condition, although, and there are some rare exceptions to this, that's a little bit unusual. What is sometimes a little harder to tease out is they're not better explained by the symptoms of another mental disorder. The example I often give is with eating disorders, where if one can have obsessive compulsive phenomena, but if they're entirely related to body image or concern about gaining weight or about food, we typically wouldn't call it an eating disorder, but there's a very high level of comorbidity as well. So if those obsessions were about food, but they're about maybe the food is contaminated, I'm going to get sick if I eat it, you might think, maybe that diagnosis of OCD is all warranted, for example. So it gets a bit complicated, but certainly if it was all focused on some other, the central concerns of some other disorder, you might not make the diagnosis of OCD. And then typically we think about the variable levels of insight with OCD, although for most people, including most children and adolescents, by the way, have fairly good insight. And I won't get into the details of this, there's some relevance as to whether they have comorbid tics as well. So OCD, just a few kind of facts. So OCD affects, quite common, affects 1% to 2% of the population worldwide. It has been identified as being highly disabling. There's some older literature suggesting it was one of the 10 most disabling conditions worldwide, according to the World Health Organization. Actually, frankly, some of those kind of large global burden of disease studies, the take home from those is more that psychiatric disorders in general are associated with some of the highest levels of disability in the population, OCD not being no exception to that. As I mentioned earlier, 55% heritable based on twin studies. median age of onset is 15. So I see entirely children and adolescents, and I'm kind of, I'm kept quite busy because that's actually when OCD begins. Of course, there is sometimes a long delay to diagnosis. So adult clinics are often full of people who actually had early onset, but it can develop into adulthood, into sort of 20s. It's unusual for it to kind of develop for the first time 30s, 40s with no preceding history. And you start to wonder about other things if that does seem to be the case. And I won't get into treatment in this talk, but there is effective treatments, primarily SSRIs and a particular variant of CBT called exposure response prevention. They're very effective, but they don't work for everybody. So there's still a lot of room to understand causal factors, pathogenic factors, like as with genetics, and develop better treatments. There is this category. I did title the talk about related disorders. I actually made a decision when I was preparing the talk that I won't get too into the other disorders, particularly as there's just not a lot of data, especially in genomics, or almost no data in most cases. We do have a bit of data on hoarding. I could talk about that maybe in the questions if people are interested. So hoarding is a OCD-related disorder, but we have basically nothing substantial on the other related disorders that fall into that DSM category. So body dysmorphic disorders or body-focused repetitive disorders such as trichotillomania or skin picking. But they do fall in the same category in the DSM, and there's a high level of overlap clinically. So genomics of OCD. So where are we? So there's a history, which I won't, again, get into, but, my group and others have been involved, and there's a history of, first sort of linkage studies. That's actually a little before my time, but there were still linkage studies happening. And then what we call candidate gene studies, where you pick a particular target and you look at that, whether it's related to OCD for various reasons. The field has really shifted away largely because those were hard to replicate. There were a lot of problems with false positives with candidate gene studies. Largely because we're just not really understanding a priori the mechanisms, the biological mechanisms for most psychiatric disorders. So we weren't probably picking the right candidates because we couldn't. We just didn't know enough. And so the approach now is to become more agnostic and use more genome-wide approaches, such as the genome-wide association study that I already alluded to. So this slide encapsulates very quickly kind of the history of genome-wide association studies in OCD. And all of these are, actually, with one exception, are ones that I've been involved in one way or another. So initially, our group published this paper, it's actually over 10 years ago now, in molecular psychiatry. And I'll orient you briefly to this whole idea of this is what we call a Manhattan plot. You're going to be seeing a few of these, so I'll just orient you to what that is. So a Manhattan plot is based on it's really based on the New York skyline. So the idea is that you have a lot of very tall buildings, and what it looks like, if you look at graphically, what they actually represent is that on the y-axis you have all these multiple common variations across the entire genome, hundreds of thousands of them, but kind of clustered together in this illustration across the different chromosomes. So that's sort of across the genomic space. And then you have transformation of the p-value, so the significance. So what you're looking at is the minus log p-value here. What you're looking at, so statistical significance for genome-wide association studies, because you're correcting for these multiple tests, is p of less than 5 times 10 to the minus 8. So not for, kind of your less than 0.05 kind of thing you think of traditionally in terms of like a single test. So quite a stringent level of statistical significance. So what you're hoping for, I mentioned that Manhattan skyline is sort of an Empire State Building that kind of sticks out over this red line of that, kind of significance. What you often get, as we did in that first study, is you get a few slightly taller buildings, like the Rockefeller Center or something like that, or, kind of thing. Kind of above that, kind of that more suggestive level. But what that really suggests is that we need larger samples, and the story of GWAS in general, and OCD is no exception to this, has been this kind of progression of getting to larger and larger sample sizes. That has its own pros and cons, by the way, as you'll hear, in terms of introducing heterogeneity and getting different kinds of samples that may be for example, less rigorously phenotyped. So I'll talk about that. So the history of this is we had two GWAS that came out around the same time. So there's one by my colleague, Manuel Mathison, who's now in Canada. It was, by the way, strong Canadian representation. So Evelyn Stewart is the first author on that paper. That first paper, my colleague is at UBC. Manuel is now at Halifax. I'm in Calgary and was at Sick Kids. So there's been a strong, when some of this work was done, so there's been a strong Canadian contribution to this, by the way. Anyway, so we had two GWAS with sort of similar kind of, sort of no no genome-wide significant findings. We did a meta-analysis, and again, nothing really. More recently, and I'll show a bit of detail on the next slide, we have, and this is available as a preprint on medRxiv, is actually under review. We're hopeful because it's our second round of review from Nature Genetics for a latest study, which is effectively a meta-analysis of multiple cohorts. And in that study, we've and I'm saying we, it's a large group, and I'm not the senior author on this, but sort of have been part of the work and contributed some of the cohort samples that have been involved in this. We've identified 15 loci from about 37,000 cases, and how do you get this many? I'll tell you, these are like controls collected for large studies from biobanks. So this massive number of controls, 949,000 controls. And as you can see here, actually, I'll blow it up here on the next slide. Now we're starting to see some Empire State Building type. This is the graph is sort of slightly different. So it almost looks like it's not as high, but it actually is. Here's the significance line. So now you're getting these 15 significant loci. Some of them are clustered together and may effectively represent the same finding. But you're starting to see this, and kind of all very rigorously good, really good quality control of the data. So we're just starting to see now, when in this largest GWAS to date, some significance. What are we finding? By the way, these are largely European ancestry samples, and I'll get to that. That's a big limitation as well of genomic studies in psychiatry in general. And I mentioned about the heterogeneity. This included the original samples were relatively rigorously phenotyped samples of, like, they'd had a semi-structured interview confirming OCD. They had standardized measures of their OCD symptom severity, et cetera, like pretty kind of what we'd expect in a more subspecialty clinical setting, for example. But then it's expanded to more like clinical registries. Some of them, because their primary diagnosis was an OCD, but they came in. Those ones actually still look like they are, we have relatively high what we call SNP heritability, which I'll get to in a minute, which suggests that we're more likely to find something. You remember that graph of like higher heritability, more likely to find significant loci. But we've also got, for example, a large component in this study of we actually use the 23andMe data. So these are like some of you may have participated in 23andMe or Ancestry or something like that, where I've done Ancestry, where you get your kit and what you get asked is like, would you like to fill out a questionnaire? And you say, have I had OCD? Yes or no. And so pretty like not, like many of those people may have had OCD, but it introduces a lot of noise because either possibly they didn't because they misunderstood or misapprehended or checked off the wrong box, or because it's only a single question, or maybe it's very mild and the genetic contribution is lower than it would be for somebody who presented to a subspecialty clinic. So you actually see this. You can measure what's called the SNP heritability. So that's kind of looking at the heritability based on your actual genetic data. So if you look at the clinically ascertained sample, it's actually pretty high. It's like 32% SNP heritability. Below, by the way, is typical, below what you get from twin studies, but not bad. Like, pretty, there's not as much, quote, missing heritability, whereas it's tiny for these like for the self-report 8% SNP heritability. So suggesting there's a big gap, and that just the sheer number may be contributing to your findings, but that there's some limitations with using this approach. And this is not unique to us. This is a lot of studies are using the same approach because of this need to to get larger and larger samples, but we're running into barriers in terms of when we do that. We did also find that OCD was genetically correlated with 40 disorders or traits, many of the ones that make sense, like genetic correlation with anxiety disorders, for example. And that correlation was where we'd want it to be. That was positive with all the psychiatric disorders we looked at. Interestingly, negatively with BMI, age, these, and we don't understand all of these, but age at first birth and multiple related autoimmune diseases. In terms of what genes, and I always hesitate to make too much of these, although now maybe I shouldn't be quite as hesitant because now that we've got multiple loci and they seem strongly significant. But so I won't get into the functional significance of this, but we did find sort of regions that were interesting. So the MHC region, so that's involved in your immune response, and there's a hypothesis that some of OCD may be related to early infection, for example, autoimmune response to early infection. So kind of you could postulate that makes sense. A gene called furin, which is involved in has a lot of functional effects in the brain, has shown up in other psychiatric disorders. And we did replicate, because remember I showed those even in that meta-analysis with a relatively large sample. kind of thing. We replicated one of our stronger hits from or the strongest hits from an earlier study in 3P21. So we were, comforted by that. And another thing so that's kind of where we're at with the genome-wide association study of OCD diagnosis. The other thing we've learned a bit is about and there's a group within so these studies, by the way, are carried in the auspices of what's called the Psychiatric Genomics Consortium, which is one of the largest, if not the largest, collaborative groups in psychiatry research in general. And that brings together all the researchers for a particular disorder or trait of interest. So there's a group, and we actually work together with the Tourette's group because of the sort of common comorbidity between those two conditions, as you'll see. There's an OCD Tourette's working group that I'm part of. And if you look, there have been studies that looked across all of these studies that are contained within the Psychiatric Genomics Consortium called the Cross Disorder Group. And that group has been able to look at what are the actual correlations, what are the common risk factors. And they've done some really interesting correlation factor analysis clustering. And interestingly, OCD seems to be the most closely related to at the genetic level with Tourette's, which wasn't a big surprise, but eating disorders, also not a huge surprise, but maybe not quite as intuitive to some people, but there's a lot of clinical overlap between anorexia, OCD, and then with Tourette's. And you can see kind of where some of the other disorders kind of cluster as well. So another approach I wanted to talk about briefly, though, just shifting away a little bit, just giving you a bit of an overview of some of the work that we've been involved in, from another approach to looking at OCD diagnosis or any diagnosis is to take a more quantitative trait approach. So that's something that we've done quite a bit of work on. And it's really based on this concept that many disorders, especially common disorders like OCD, and this is we've shown from some of our own data using a large community-based sample that I'll talk about, of children and youth, is you've got this normal population variance in the variation in symptoms, and then the tip of the iceberg is your clinical disorder, based on sort of this distribution, which kind of makes sense, and there's some evidence to support that. So therefore, but the genetic risk, of course, may also contribute to what's below the surface. So using a quantitative trait approach has a number of advantages, some just straight up statistical advantages, so the greater power of doing quantitative trait analysis versus looking at a categorical trait. So we developed this study that's now, we've stopped recruiting. Sort of interesting story. It's called Spit for Science. The study takes place, and my colleagues who are most instrumental in that are really carrying this, really leading the charge on this. Jennifer Crosby, psychologist and senior scientist at Sick Kids, and Christy Burton, a research associate there. Also, Russell Shacker has been a key player in that. This is a study that's actually where we went to a science museum and asked people to participate in this research. And recruitment has stopped at 40,000 kids. Much of the results I'm going to talk about there's sort of in two waves. And the first wave was of 17,000 kids, only about 5,000 to 6,000 that we genotyped initially. So a lot of the results are based on a small subset. So there's more to come because we haven't genotyped, only genotyped a small proportion so far of these 40,000 kids. And what we did is we got physical, mental health phenotypes, which included just asking them more self-report kind of questions, but also doing more quantitative trait measures, which we paid a bit more attention to. We got DNA from saliva. We did get, and I'll talk just a little bit about what we're trying to do with that data, some environmental risk data based on their neighborhood, where they lived. We are starting to do some work. I won't talk about this at all because I don't have any data. And I've been less intimately involved in this. It's been other investigators have done this, looking at the oral microbiome, which is kind of interesting. And then a little bit of information about demographics, lifestyle. So the interesting story of this was, it was an interesting endeavor. I mean, so we're asking kids to, sometimes we call it score, stop, and spit. So scoring is they score their measures. Stopping is they do this cognitive measure called this stop signal task. It measures inhibitory control, and then we get them to spit and provide saliva. The science center literally shut down. Now, you probably heard this story kind of thing. They had some structural problems. The science center, within months after we stopped recruitment. So it's sort of fortuitous that we kind of stopped when we did. It was a difficult study. We had a big gap during COVID when spitting in a public place was not a good thing to do, let alone gather in a public place. And but we sort of persevered, and we were able to get up to that number, which was pretty close to what we'd originally planned. And there's open consent, so data will become accessible via the Sick Kids, Healthy Kids Biobank. We use this quantitative measure that we developed for the study, partly because we weren't satisfied with the the typical clinical measures that tend to be much more skewed. So you get a lot of zeros. So we developed this, we attempted anyway to make it more normative. Linda and her group are translating into French, currently just available in English. And it's a 21 item measure, was kind of based on the other clinical screening measures. And we have done some heritability within our sample, partly because we had a very, because 40,000, we had we did have some twins. So we looked at twins, and we also just looked at, we did sort of an estimate of heritability. And it's actually pretty high for the total score. So it's been now psychometrically validated in the clinical setting and has shown good sensitivity and specificity for OCD. So that's kind of the basis when I show my results. What we're basing this on is this is our quantitative measure of OCD in the general population. So we did publish on this a few years ago. Again, sample size related, although we have high heritability, we still had for a GWAS study, relatively modest sample size, although it was about 5,000 kids, I think it went into that. So we did have a genome-wide significant association. We were able to replicate that in some case control samples available to us at the time. So that specific variant gave us some, not strongly significant, but at least that variant did replicate and was statistically significant. And it was a gene called PTPRD, which is involved in synaptic transmission. And there's been a lot of interest in psychiatry and psychiatric genetics about the role of sort of synaptic functioning, and then a lot of genes that have shown up for neurodevelopmental disorders, for example. There is and I'll touch on this, some of our more rare variant studies of OCD have shown this gene called SLITRIC, which is also kind of in that sort of that system, the sort of postsynaptic complex. And we wondered about our population traits the same as clinic, so we wanted to look at polygenic risk, and for OCD and sort of look at the comparison between our clinical and community-based samples. And we did in fact find that. So what this graph tells you is basically, if you're predicting the quantitative score using polygenic risk scores based on the clinical data we had on the clinical GWAS we had available at the time, you do get quite a high prediction or predictive power of the polygenic risk score based on OCD. If you look at OCD as your discovery sample, but a little bit, but less so for other disorders. A little bit for anxiety, which kind of makes some sense, but really not very much at all for ADHD. So there does seem to be some specificity in that. We've gone on since then and recently published a genome-wide association study of obsessive-compulsive symptoms in a large international collaborative sample. So this included, so it overlaps with the study I just talked about, because it does include data from the Spit for Science study. But we brought together other groups that had quantitative OCD traits. The total sample size pooled together 34,000 individuals, and including and again biased towards European ancestry. We did encompass both adult and child, so there's one source of heterogeneity, and there are others in terms of the questionnaires themselves. But that was sort of based on the available data. We wanted to get a large sample size, so we brought together, and the other studies are mostly twin studies, by the way. And our aim was to identify novel risk variants for quantitative OCD traits and look at shared genetic risk, similar to the aims of the Spit for Science study. And our key results, we didn't get a significant genome-wide. You think, well, huge sample size. But again, we hypothesize that some of the differences are, introducing the heterogeneity in terms of, we think that adult onset OCD, for example, doesn't have as strong a genetic contribution, so we probably might have lowered our heritability. Heterogeneity in the questionnaires, in particular, many of these studies did use I mentioned the kind of more skewed questionnaires of screening for clinical disorders, so they didn't get they weren't capturing the full distribution like our tox measures did. They just didn't do it that way. I'm not sort of tooting our horn. That's just the approach we took. We did, though, find when we looked at, there was some interesting, and we'll see how this bears out. It is a little bit consistent with some other data that we've seen. We did do some sort of post-GWAS analyses. So we did, in our gene set analysis, interestingly, we did find a gene set that was implicated in insulin-mediated glucose disposal, and that did reach significance. So gene set analysis, you take a group of genes that are functionally related. And they're interesting. Some secondary data analysis that we were involved in previous GWAS studies did suggest that dysregulated insulin signaling had been implicated. So that might be a genetic pathway. Again, I think too early to say for sure, and the PRS for OCD diagnosis was associated with OCD, obsessive compulsive symptoms, and there was a substantial genetic correlation. So if we take the OCD case control and the OCD quantitative traits of like genetic correlation of 0.72. So we were comforted by that. In other words, we feel like, okay, we're measuring a trait that the genetic basis correlates with the disorder that we think it relates to. So I think that's kind of important and reassuring. So it does sort of support the hypothesis that OCD represents the extreme end of a widely distributed obsessive compulsive symptoms in the population. I won't talk. How am I doing for time, by the way? Yeah. Okay. So I'll just touch briefly on rare variants, just to talk about that. So because I wanted to leave a bit of time to talk, but then I'll open it for discussion around this idea of like where do we go from here, more precision medicine approaches. But so there has been a little bit of work in OCD and rare variants. So for example, rare copy number variants. So these are where you have chunks of DNA basically that are either deleted or inserted, typically greater than 1,000 base pairs in length. We did do some earlier work. Our group and others found an overall enrichment of copy number variants in genes implicated in neurodevelopmental disorders in OCD, and there have been some recently what we call whole exome sequencing. So this next generation sequencing approach. You can identify specific variants, but because you're covering the whole genome, you can identify things that maybe aren't found in the general, like in using these common variant approaches. The largest of these two studies, there's been a family-based study and a larger one that included both family-based and case control samples, did find that there was an interesting result for a single gene result for single nucleotide variants within a gene called SLTRIC5 that kind of falls in that pathway with PT or PRD that I mentioned earlier. So kind of interesting. There'll be more work done in that area. This is just a repeat of what you saw earlier. So I wanted to just briefly talk about how do we sort of integrate this with social environmental determinants, and then talk a bit about sort of precision medicine. First of all, it's important to point out that, again, one of the things, so what are some of the gaps and what do we want to look at? One of the gaps and one of the areas that by collecting a more diverse sample, both in terms of the socio-environmental background, but also ancestral background, is you see one of the worries that has raised some alarm and caused a lot of discussion in groups like the International Society of Psychiatric Genetics is this idea that using current polygenic risk scores may actually make health disparities worse, not better. Because so for those of us in the room who are of like and I count myself among those, I know we're a diverse group, but who are of like primarily European ancestry, like I could benefit because if you looked at polygenic risk for a disorder for me, it might tell you something. The predictability might be that high because, as you can see, so much of the data is based on people are more closely related to me genetically. But not so much for other populations. You can see here, this is a really nice slide of the predictability. So you got this narrow band of like, now this is partly because it's intentionally meant to be a benchmark. And that doesn't mean it's 100% predictability for Europeans, by the way. But if you benchmark it against Europeans, you can see that it slowly goes down if you take a broad American population. But then if you go down to like, people of African origin kind of thing, ancestral origin, really poor. And again, it's because there's not much data on people who are ancestrally related to them. There's a lot of efforts to correct that, but we need to do more of that in future. By the way, we do have a fairly different, that spit for science sample that I gave. I think once we start looking at the larger sample, we'll be able to get at some of that because we do have a more heterogeneous population in terms of ethnicity in some of these studies. We're also starting to look at variables, particularly neighborhood level environmental variables that we can get from just knowing where people live. For example, you can derive measures of what's called the material deprivation index. So you can benchmark postmarks using this CANU data set, postal code against census data. So you get at things like their socioeconomic status and things like that. And we're starting to look at that. This is the Metro, probably recognizes a map of Metro Toronto, but you can kind of look at where do people come from, and what level of material deprivation we're starting to, look at. Now there are sample sizes larger, thinking of doing, more genome by environmental interactions. So I want to just talk briefly about, and before closing, and want to allow enough time for questions about kind of where are we going with all this? That kind of idea of a pathway to precision medicine. Well, current practice, as you know, is kind of for psychiatry disorders. You see someone does a clinical interview, they benchmark it against the DSM, and they they provide a diagnosis and they provide a range of treatments. And that works quite well, but it not well enough. And so I think where our vision in the future, is that we can do a lot more if we include some biomarker information, in this case, particularly genetics. It's sort of overly simplified idea, but the general idea is there that, we can do things like staging people, like identifying based on their genetic profile, like how likely is it to develop more severe illness or maybe develop a comorbid condition down the road. We can target our treatments better. And in particular, an area where we're starting to get some traction, there is a little bit of evidence we can already target some of their choices in terms of medication. So I'll skip over this because I wanted to get to that. So we're doing a little bit of work. And again, this is adjacent to our OCD work. So this is work in Calgary. We have a study called PGX Spark. So psychiatric, so pharmacogenetic guided prescribing for kids. And the idea is that any child who's starting a medication or switching to a different medication can get basically get genotyped on a panel of pharmacogenomic related variants for free as part of the research study, and then they get the results back to them and to their physician within 2 weeks. And so it's an implementation study to see if we can get this to work. Our idea is to leverage this to really kind of like introduce precision medicine using genetics into the clinic. And it's based on this concept that, at the group level, these medications work on, 50, 60% of individuals, but at the individual level, there's a lot of things we don't know a priori when we chose a particular drug for an individual, and they could fall to any of these boxes. They could, ideally, the medication is highly efficacious over here, highly efficacious and they also tolerate it. They don't have side effects. But if they fall into one of these other boxes, they may not do as well on the medication, and we can use genetic information to inform that. So we've developed this study, which involves Canada's first evidence-based pharmacogenetic testing service in child mental health, we believe. And we've now kind of, the study began in Alberta, and that's still where bulk of our recruitment has been, but we've been kind of spreading out to within Western Canada. We're, applying for some funding to potentially branch out and do this even more broadly, but that gives you an idea of the uptake has been really, really good. We started this during COVID, which interesting. We learned some things that may end up being useful in the end because we started during COVID. We basically mailed out kits, and because we had to mail out kits to people's homes, we thought, well, we don't really have to restrict it to subspecialized clinics where we have a better chance of seeing them in person and having the time to spend with them to consent them. We could just send it, we can enroll them through their family recruit them through their family physicians, pediatricians, psychiatrists in the community, et cetera. So they get the information back about their pharmacogenetic profile that they can use in the form of kind of a little wallet card or passport kind of thing. And because it's their sequence and it doesn't change, they have that for the rest of their life. But in many cases, it does improve treatment. And we've done just a little bit of work on this, showing that because there's not a lot of data in kids, we just wanted to ground this. So looking at effect of like cytochrome. So these are mostly these metabolic genes that influence, kind of how your drugs metabolize in your body, and therefore affect the levels and side effects and as well as response theoretically. And so we've done a paper on this led by Chad Bousman, my colleague, by the way, so shout out, who's leading this work, and a trainee working with him, looking at the impact on of fluoxetine and whether we can predict side effects and symptom improvement. And sure enough, we found that there is we do find a good correlation with their pharmacogenetic profile for cytochrome so for the 2D6 gene, which is involved in metabolism of fluoxetine particularly. Globally, by the way, like about 75% of individuals will have something on this panel that's potentially actionable. Typically it's in one of two genes. And part of what we're trying to demonstrate is like, it's potentially actionable, but does it influence their treatment, and B, what's the uptake of that? Like, is it so much like, we can't really dictate to the physician receiving this information that they have to change the drug, for example, but we're studying as to whether they actually do that and whether that's useful in practice. And we're collecting health administrative data. We've also got a trial, and again, I think I'm probably getting close to time, but we do have we are starting to have a number of other trials sort of thinking about where we're integrating genetic information. So we have a cohort trial that's begun, what's called a cohort study that's based on this model of a master observational trial funded by the Ontario Brain Institute. This is in collaborations with the the lead site is CAMH. There's 5 Ontario sites. So I'm one of the co-leads, and we have collection in one non-Ontario site, which is in Calgary. Our aim in this study is to look at help-seeking youth. So we're hoping to recruit a total of 3,200 kids, and kids, broadly speaking, because it does go into that emerging adult age range, so 11 to early 20s. And they will have a common phenotyping protocol. Basically a sort of electronic questionnaires, permission to link to electronic health data. One of the complications we haven't fully worked out is because there's 2 provinces involved. It's a good challenge, though. I think one we do need to work out, but like, how do we, figure out an effective way of sharing and keeping data consistent? And then there'll be a sub cohort that'll have somewhat more deep phenotyping, things like neuroimaging and using basically using phones to kind of monitor for with consent, sort of for self-monitoring and that sort of thing as well. And then the idea is embedded within the master observational trial model is that you get this rich set of data. It was sort of developed in areas like cancer on a very large cohort of individuals, but then they can be selected for specific trials. So we're just sort of beginning that process. This study is literally just launched as of like this fall. And so, yeah, I'd like to acknowledge, others who, folks who did a lot of the work. I'm representing, kind of very large collaborative projects. I'm certainly missing some people, but key contributors or collaborators were in the Spit for Science project that I mentioned earlier. I've mentioned my collaborators there already. Folks at University of Calgary, sort of in my group, and in Chad Bausman's pharmacogenetics group, work very closely together. Some of the OCD quantitative work involved Sandra Meyer and Manuel Mathison, who's at Dalhousie. And other colleagues in Europe, Dirk Smit, Norrstrom, and others, David Mateix, Coles. So yeah, I'm happy to take any questions. Anybody wants to visit Calgary? That's kind of not that, that's not Calgary, but it's like an hour or so away. So beautiful place to visit. Love to see some of you, and thanks again for the opportunity to come here as well and present to all of you today. Yeah.
Thanks a lot for this excellent presentation. I really like an impressive amount of work. So we have 10 minutes for questions.
So
you mentioned some genes, very interesting. Like what did they actually do?
Yeah. So no, I skipped over that relatively quickly. So it varies with the study. So in terms of the one of the key findings that we found, it was actually probably our top hit in terms of a specific genetic locus, was the MHC complex, interestingly. So with the and that's being identified in other psychiatric disorders like schizophrenia, for example. And so that's probably that's sort of our top hit where we actually know something, because we have a locus where we actually don't know what it does, other than like there's no clear candidates within the locus. The only thing that we have observed is that it also has been identified in other psychiatric disorders. So there could be something there and probably requires further investigation. But yeah, the MHC complex is the one in the OCD diagnosis, large GWAS, that seem to be the strongest finding. And again, hypothetically, it could support, an autoimmune contribution to OCD that's already been hypothesized. There's a subset of kids with OCD, for example, who we know, I think it's safe to say we know there's a little controversy about how we identify those kids and how we treat them, develop OCD, interestingly, abruptly, suddenly, following a streptococcal infection. It's this phenomenon called PANDAS, pediatric acute neuropsychiatric disorders associated with strep. And so could be, and it's been hypothesized that, they're the obvious ones where there's a clear link, but there may be a broader group of individuals with OCD. There's some epidemiological work suggesting that higher rate of childhood infections and people, have like largely the Scandinavian registry studies have sort of suggested that in their early childhood. So that's kind of where we think that's maybe, to me, was one of the most exciting ones. The other one that's come out of the quantitative trait studies, which is interesting, but I think I'm even more cautious because it's based on a post hoc, gene set analysis. I think it would be nice to see it replicated and where we have a really solid, statistically significant finding for at least one locus, is this sort of insulin signaling hypothesis. And so that's also, and there's, a little bit of work suggesting there was that kind of negative correlation with BMI. There could be a relationship. There's the correlation with eating disorders, where a lot of metabolic genes are thought to be involved. So these are very broad hypotheses, but that's sort of what some of those systems do. And so, but we're a long way from being able to translate that into, novel treatments or that sort of thing. But hopefully we'll get there. Yeah. Yeah. Leonard.
Thank you, Paul. Very enjoyable. Thank you.
Yeah, thank you.
We don't get to hear a lot about OCD. It's only having to get these talks, so I really enjoy it. One question I had in my mind is when you talk about irritability, we often give a number. Some people say 80% and so on. But often it changes with age as well, right? So with OCD, for example, do you know anything about does irritability change with age? Is it right? And I think what I'm getting at is there genetic structural differences between adults and child OCD?
Yeah, we do know a bit about it. It's a really good question. First of all, just the study of OCD, the heritability is largely based on quantitative, just to sort of show where the data is coming from. It's largely based on quantitative report data kind of thing. So there have been some smaller studies of actual individuals, like one person has clinically diagnosed the OCD, their twin does not, that sort of thing. But this is primarily based on quantitative traits, and you get the sufficient sample size kind of thing that you want. In terms of age, what we do know is that if you take a group of individuals, our sample included, by the way, who were ascertained in childhood, and you do heritability analysis, you certainly do get a significantly higher heritability in children versus if you take a similar sample of adults. So there does seem to be this age of onset. There's some hypothesis in that because if you actually look at it's quite interesting. If you look at the distribution of age of onset OCD there, and this is debated a little bit, there may be a bimodal onset as well. So it actually might be a distinct group that has a stronger genetic contribution, and that's what's being reflected in those childhood onset twin studies versus the adults, where it actually looks a little more like major depression, for example, in terms of the levels of heritability. They're more like in the 30-ish% range, kind of 30 to 40%. And there's a greater comorbidity with depression, that sort of thing. So there's a variety of reasons to think maybe that kind of makes sense, but so there are some differences. It's a really good question. Yeah.
I'm monitoring the chat. There's no questions in there, but this is mine. I was wondering if there's any differences in prevalence for sex, males versus females, and in the heritability, any SNP kind of effects, and then towards the position of medicine if sex effects is good.
Yeah, it's a great question. As we delve into the questions, we know less and less, but what we do know is that the sex ratio is roughly even if you go across the board. Interesting, it kind of builds on Lena's question actually, is that in childhood, so early childhood, particularly prepubertal samples, you get more boys with OCD. And that may reflect, they also have a higher comorbidity with Tourette's, ADHD, kind of more of a neurodevelopmental picture. So that kind of early onset group, especially quite early onset, like before age, sort of age 10, before puberty, you're more likely to be boys. And that's, by the way, somewhat true across the board in those people who work in child psychiatry will, see this, you get more boys in your clinic on average. Partly it's because of the ADHD, for example, a bit more disruptive, et cetera, than girls. But there is probably a phenomenon there of increased prevalence. Then as you get into adolescence, it starts to shift, and you get it for adult onset OCD. It's a little bit more of a slight predominance of females, so female sex. But if you look across, then it sort of evens out. So you get a roughly 1-to-1 ratio, as opposed to, let's say, neurodevelopmental disorders like ADHD, where you get quite a strong male predominance, or affective disorders, depression, anxiety, later in life, where you get, typically later in life, I mean, a little, late teenage or early adulthood, but you get you tend to get a female predominance, quite a high ratio. So it seems in OCD is kind of an interesting disorder because it sort of washes out those effects because of that continuum. You'd asked about, can't remember the second part of your question. Yes. Yeah. So So that's where I was sort of saying, like, I think we know less and less. So in terms of heritability, there may be, and some of the data suggests there might be a higher heritability in males, but it's totally confounded by the fact that it's that onset, that age of onset. So I don't know if I trust that. I don't, in terms of whether heritability, it feels strongly that heritability is higher. We haven't done enough about sort of sex specific effects in terms of SNPs. We don't really know. And it's members of our group have sort of done things sort of in a preliminary way, but I think we've been hesitant to really explore too much in that area just because the sample sizes have been, you're effectively splitting the sample and so the samples have been small. I think now that we're approaching these sort of large, and we're at these larger samples, I think we could start to do that. And I think would be really interesting because one would hypothesize for sure there could be some sex specific effects. There are some sex specific effects in clinical manifestations of OCD that are kind of interesting, for example, that again could end up being reflected in the genetic data. So there is for sure pretty good evidence of potential hormonal effects, particularly during changes in the reproductive cycle with women. So common times when OCD can either flare up or just appear in one case. So, there's certainly a phenomenon of kind of like a young women developing OCD for the first time around puberty, or then their symptoms being associated with menarche, with their menstrual periods, but flaring up for the first time with menarche. And there's also, then this is really quite well described, is like around pregnancy. There's a subset of women who will develop frank, full-blown OCD kind of during the peripartum period. It's actually kind of interesting. Sometimes the intrusive thoughts are about, by the way, most, I know, kind of speaking, sort of what, one step removed kind of thing from my wife going through this kind of thing. Like, it's quite common for one to get at least some intrusive thoughts like, oh, what if something bad happens to my baby? But this is where it gets really bad, where it kind of like really takes over and becomes quite harmful. And they meet the full criteria for OCD kind of thing. And there's a phenomenon there. So we do think there could be a hormonal link, and possibly, one wonders if that could also be reflected in the, genetic expression as well.
I just had a clarification question.
Yeah.
On the slide that you were seeing, or like showing the predicted value of GWAS with like looking at different, like people of different ethnic origins. I think one of them said like American.
I glossed over that. So that is kind of a mixed population, and they were doing that intentionally, where the ethnicity is either unknown or assumed to be kind of sort of mixed background, as opposed to people who are known. Like, I mean, the strictest definition would be, sometimes it's just based on from it, which is probably not a safe assumption now, but it might have been at one time if their origins are from a European country. But more typically it would be like you have some evidence that they say, we did this with Spit for Science. We asked, where did your grandparents come from, kind of thing. So you've got at least a pretty good picture of like their ethnicity. So that was the reason for that, and that's why it kind of falls in between. But it's a little closer, but there's also huge error bars because it is so mixed as opposed to that.
Did you know the makeup of that American group, or was it?
I'd have to go back. That's a really good question. Because that was like based on that wasn't our data, but I'd have to. That's a really good question. I have to go back and look at it. Whereas the others are more clearly defined based on their ancestry.
Well, thanks so much for coming. And
yeah,
it's great talk.
Yeah, thank you all. Yeah.